Dear friends, hearty welcome once again. Friends, all of you are known. In our previous lecture, we have started the study of major area of this chapter, that is post-fertilization changes. That is the changes taking place in the ovule and ovary after the process of fertilization. We all are aware, after the process of fertilization in angiosperms, the ovule is transformed into the seed and ovary is transformed into the fruit. So, the development of seed from ovule and development of fruit from ovary are nothing but the post-fertilization changes. Now, already we people have completed the study of first post-fertilization change, that is development of seed from ovule. And here we people have to study about the development of fruit. I said just now, just like the ovule, after the process of double fertilization, the ovary wall also undergoes certain noticeable changes, and thus the ovary is converted into the fruit. Actually, the fruit is defined as ripened ovary. The fruit is defined as ripened ovary because after the process of fertilization, the ovary undergoes certain quantitative and qualitative changes. Quantitative in the sense, I will say, certain physical changes, certain chemical changes, and of course, those changes taking place in the ovary wall which bring about transformation of ovary into the fruit are known as ripening changes, and thus the fruit is defined as ripened ovary which may or may not contain seed. Now we people have to study about that ripening changes. First of all, we must know that the transformation of ovary into the fruit, the conversion of ovary into the fruit is under the influence of stimuli received by ovary from the hormones released out by pollen tube and also the developing seed. It is believed that when pollen tube containing male gamete enters into the ovary cavity, releases certain plant growth hormones into the ovary cavity. Listen carefully. I said that when the pollen tube carrying male gametes enters into the ovary cavity, at that time that pollen tube releases certain plant growth hormones into the ovary cavity. Plant growth hormones, you know, these are complex organic substances which play important role in the plant growth and also many other, we can say, metabolic activities, physiological processes of the plant body. There are actually 5 different major groups of plant growth hormones that we have to study in separate chapter, plant growth. But temporarily I would like to tell you, there are 5 major groups of plant growth hormones such as auxins, gibberellins, cytokinins, ethylene and abscisic acid. Now I said, whenever the pollen tube enters into the ovary cavity, at the time of fertilization, this pollen tube releases certain plant growth hormones such as mainly auxins, gibberellin, cytokinin. In the same manner, the developing seed within the ovary cavity also releases some plant growth hormones, same such as auxin, gibberellin, cytokinin as well as ethylene, and it influence or it induces the ovary wall to undergo different qualitative, quantitative changes known as ripening changes, because of which the ovary wall is converted into fruit wall. तर आता मी असं तुम्हाला म्हटलं की ज्या वेळेला पोलन ट्यूब ही ओव्हरी कॅव्हिटी मध्ये मेल गॅमेट्सला घेऊन प्रवेश करत असते, त्यावेळेला ही पोलन ट्यूब ओव्हरी कॅव्हिटी मध्ये काही हार्मोन्स रिलीज करत असते, ऑक्झिन, सायटोकायनिन, जिब्बर्लिन या गटातले. एवढंच नाही तर डबल फर्टिलायझेशन झाल्यानंतर ओव्हरी कॅव्हिटी मध्ये ओव्ह्यूल पासून सीड डेव्हलप होत असताना ही डेव्हलपिंग सीड सुद्धा वेगवेगळ्या प्रकारचे हार्मोन्स हे ओव्हरी कॅव्हिटी मध्ये रिलीज करत असते आणि या वेगवेगळ्या प्लांट ग्रोथ हार्मोन्सच्या परिणामामुळे ओव्हरी कॅव्हिटी, ओव्हरीच्या वॉल मध्ये काही बदल होतात आणि ओव्हरी वॉलचं रूपांतर हे फ्रूट वॉल मध्ये होत असतं. त्यांना आपण रायपनिंग चेंजेस असं म्हणतो. नाउ इनिशियली that different plant growth hormones they stimulate the rapid cell division. They stimulate the rapid cell division of the cells of ovary wall. Cells of ovary wall, and thus it occurs increase in the thickness as well as size of ovary. Listen, I said that initially the different plant growth hormones they bring about the rapid cell division of the cells of ovary wall, due to which the ovary wall subsequently becomes thicker and thicker, and also it leads to the increase in the size of ovary. After the sufficient growth of ovary wall, we can say now it is fruit wall or it is fruit. After the sufficient growth of fruit, it leads to the actual ripening changes, actual qualitative changes. That ripening changes we have to understand properly. So let us talk about that qualitative changes or ripening changes. Ripening changes. First of all, it occurs the softening of the fruit wall. Softening of the fruit wall. The softening of the fruit wall takes place due to the degradation, enzymatic degradation of the middle lamella of cell wall of the cells of the fruit wall. Fruit wall तयार करणाऱ्या ज्या सेल्स आहेत त्या सेल्स मध्ये असणारा जो मिडल लॅमेला, आपल्याला माहिती आहे मिडल लॅमेला it is a layer between the two cells which bind the two cells, that clear. Now whenever it occurs the enzymatic degradation of the cell wall, ultimately it leads to the softening of the fruit wall, because you know it loses the rigidity, and hence first of all it occurs the softening of the fruit wall. Also it occurs the solubilization of the content of the cells. Solubilization of the content of the cell. And because of these two major things, that is first the degradation, enzymatic degradation of the middle wall of the cell wall, and second due to the solubilization of the content of the cells, it leads to the softening of the fruit wall. Second major change, that is the hydrolysis or breaking down, degradation of stored and complex materials complex materials in the cells into the soluble sugars, especially in sweet fruits such as grapes, mango, etc., apple, and also maybe into different organic acids, isn't it? Say in turmeric, or we can say in even lemon, etc. Anyway, so second change, that is the hydrolysis, breaking down or degradation of different stored and complex materials present in the cells into the soluble sugars. Of course, it imparts a sugary taste, that is sweet taste, to the fruits, and in some fruits there may be the formation of organic acid which impart other taste. Now come to the third major change. तिसरा बदल त्या ठिकाणी, the conversion of chloroplasts into the chromoplast. Listen, the conversion of chloroplasts present in the cells into the chromoplasts. We know that chloroplasts, that is green plastids, which contain chlorophyll, mostly chlorophyll in abundant quantity, and chloroplasts are mainly responsible for photosynthesis. But the chromoplast means these are colored plastids, and here the chromoplastids mainly contain the color pigment, that is the carotenoids and anthocyanins such as carotene, xanthophyll, anthocyanin, etc. Now here it occurs the conversion of chloroplasts into the chromoplast, which contain large amount of carotenoids and anthocyanin, which again gives a particular color to that fruits, and it is necessary to attract the birds that we are known. Again, one can say there is accumulation of different flavoring agents. Because of these flavoring agents, the fruits get a particular flavor, we can say aroma. Okay, so these different changes are actually known as ripening changes, and after ripening changes there is a formation of ripened ovary, that is nothing but the fruit. Now the fruit wall is known as pericarp. In 11th we people have studied. The fruit wall is known as pericarp. Fruit wall ला pericarp असं म्हटलं जातं. Now this pericarp may be either thin, papery, or maybe it is thick, it may be fleshy, it may be hard, it may be rigid, etc. But when the pericarp is thick and fleshy, if pericarp is thick and fleshy, then it is further differentiated into three different layers. In 11, while studying the structure of typical fruit, we have considered and studied about the mango. Let me explain you once again. Say here is the mango. Now in this mango, the pericarp or fruit wall is quite thick, fleshy, and thus it is differentiated into three layers, such as the outer covering is known as epicarp, middle juicy part, edible part, middle juicy or edible part is known as mesocarp. Here is the mesocarp. And innermost is endocarp. And of course, the seed is enclosed within the fruit. Here is supposed to be the seed. In this manner, the fruits in which the pericarp is quite thick and fleshy, it is differentiated into outer epicarp, middle mesocarp and endocarp, inner endocarp. Now, what are the advantages of seeds and what are the advantages of fruits? That we have to study immediately. But here, first of all, we have studied about the development of fruit. Now, let us talk about the advantages or significance of seed and fruit. Significance of seed and fruit. The seeds are considered as an efficient propagule. It means the seeds are considered as most important efficient vehicle for the propagation. It is the means of propagation. Actually, the formation of seed, development of seed, and it is surrounded by fruit. It is one of the most, I will say, special feature seen in angiosperms, because of which only angiospermic plants have dominated the entire earth as compared to the other plants. Now we have to consider why the seed is considered as an efficient propagule and which are the special characters, which are the special features seen in seed. Consider one by one each. First one, seed dormancy. Seed dormancy. Seed dormancy means it is the inability of viable seeds to undergo germination even all the favorable conditions are available. Seed dormancy means it is the inability of seeds to undergo germination though all the environmental conditions are favorable. सर्व environmental conditions जरी favorable असल्या तरी सुद्धा सीड्स जर जर्मिनेट होत नसतील तर त्यांच्या या अवस्थेला सीड डॉर्मन्सी असं म्हणतात. It means after the formation of seeds for some time the seeds and embryo remains dormant. Embryo remains inactivate and it immediately does not shows its further growth, development and formation of seedling. This ability is known as seed dormancy. So I said that seed dormancy means it is the inability of seeds to undergo germination though all the favorable conditions are available. And of course, because of seed dormancy, the seeds can be transported from one place to another place, which facilitates the wide spreads and propagation of plants to the new area. Second, viability. Seed viability. Seed viability means simply the ability of seeds to undergo germination after a particular time span of dormancy. Viability means it is the ability of seeds to undergo germination after a particular period of dormancy. सीड्स काही काळ डॉर्मंट राहिल्यानंतर, सीड्स काही काळ इनॅक्टिव्ह राहिल्यानंतर, अक्रियाशील राहिल्यानंतर, त्या विशिष्ट कालावधीनंतर, त्या डॉर्मन्सी पिरियड नंतर पुन्हा त्या सीड्स जर्मिनेट होतात, पुन्हा त्या सीड्सचं बीजांकुरण होतं आणि अर्थातच न्यू सीडलिंग इज फॉर्म्ड. This ability is known as viability, and such seeds are known as viable seeds. Such seeds are known as the viable seeds. So the second major feature seen in seeds which make that seeds efficient propagule. Third one is reserve food material. Reserve food material. We know that in seeds the reserve food is present either in the form of endosperm if the seed is endospermic, we have studied initially, or maybe in the form of cotyledon. This reserve food either in the form of endosperm present in cotyledon necessary for growth of embryo and development of new seedling. So it is also one of the important feature. Next one, that is the protective coats. Protective coats. We have already studied that mostly the seeds are covered by means of two protective coverings such as the outer testa and inner tegmen. So the testa and tegmen, that is the seed coat, they protect the embryo from different mechanical or we can say different physical factors, even the fluctuation in temperature, any adverse environmental condition, and so that the embryo can survive for a long duration and overcome the different adverse environmental conditions also. Dispersal. Over deposition of seeds at one place inhibits the seed germination. Over deposition of seeds at one place inhibits seed germination and also it inhibits the growth of new seedlings. याचा अर्थ जर असंख्य सीड्स एकाच ठिकाणी डिपॉझिट झाल्या तर पर्यायाने त्या ठिकाणी सर्व सीड्सचं व्यवस्थित बीजांकुरण होणार नाही, जर्मिनेशन होणार नाही आणि त्यांच्यापासून निर्माण झालेले ते न्यू सीडलिंग्स यांची पण वाढ होणार नाही. And that's why that seeds are to be transported to different area, we can say distant area. It means the dispersal of seeds is necessary, and for that purpose naturally the seeds may show some special devices. That is, some of the seeds show some pappus calyx, some of the seeds may show some wing like structure so that they can be easily blown along with the wind. Some of the seeds may show some spiny structures so that they can easily adhere to the body of animals and birds and to be carried to the distant area. One more thing, the seeds, they can be carried by means of different birds or animals. And now, after the study of these important features seen in seed which make them efficient propagule, little bit let us talk about the fruit. First of all, we can say that fruit, it provides nourishment as well as protection to the seed. And second thing, that when the fruits are consumed, eaten by man or any other animals and birds, the fruits can be easily digested, but the seeds present within the fruits that cannot be digested and egested out along with excreta. So along with excreta, that seeds are thrown out and they get dispersed. So it again helps for the dispersal of seed. In this manner, the seeds and fruit, they togetherly help mainly for the propagation of plant body, and thus they are considered as one of the important tool for the same. So here we have completed this topic, that is post fertilization changes. So let us stop here today. Thank you.